This video is an introduction to bioinorganic chemistry, and a fun way to start thinking about metallic elements in biology is to take a blank periodic table and identify the metals that you already know that are important physiologically and to identify the metals that you already know are toxic. I'd like you to take the blank periodic table that is attached to this video in your Blackboard site and identify the metals that are important to biology and identify the metals that are toxic and point out the overlap between the two classifications of metal ions. You should pause the video now and complete this activity. Here's a periodic table that shows the metals that are used by humans and all other life forms on Earth. The macronutrients are in pink and the micronutrients are in green. These are the elements that we know are essential to biological systems. Here's another view of elements of biological relevance. In green, you have bulk biological elements. So these are the ones that we mostly study in organic chemistry. The elements that make up our proteins, our DNA, our cell membranes, and some of the really common electrolytes like sodium, potassium, magnesium, calcium. The ones that are outlined in blue here are essential for bacteria, plants, or other animals, not necessarily humans. And in kind of an aqua color are the ones that are essential or possibly essential for some species. The ones that are in this peach color are metal ions that are not necessarily essential to biological systems, but are relevant to us because they're used in medicine or in diagnostics. Being an element of biological relevance does not necessarily mean that it's used by biological systems in the wild or in the native biological system, but it might be something that's used to improve human health or used as a tool to probe human health. In yellow here, I've highlighted the elements that are known to be toxic to humans in small doses. And in orange, I've highlighted ones that are toxic in high concentrations. These are metal ions that are necessary for biological systems to function, yet are also toxic in high concentration. Now, for the elements that are essential, there are some issues that biology has in getting these elements from the environment. The first problem is lack of bioavailability, which means that these elements are not necessarily readily available to biological systems. If we look at the concentrations of the 5 most essential elements in seawater, notice that they are much, much less concentrated than they would need to be in your blood. So organisms need to figure out how to bioaccumulate these elements. This problem is really that we use these micronutrients at much higher concentrations than they are available in the environment. This table is based on real data and shows several of the most common trace elements. Note that the enrichments for some of the elements in this table are 1,500 to 20,000 times that of seawater. Iron in particular is problematic. Almost all iron on the surface of the Earth is in the form of insoluble rust. This was not always the case. In Precambrian times, the start of photosynthesis pumped a lot of toxic oxygen into the atmosphere, which oxidized all of the iron too. Iron too was much more soluble, and this oxygen made iron much less available for living systems. This was also at a time when there was a huge round of extinctions related to this evolution of oxygen in the environment. So biology had to evolve methods of capturing this non-bioavailable or insoluble iron-3. And we're going to discuss this when we discuss metal transport and storage. This takes a lot of biological effort to do. Bioaccumulating these elements is hard. So we've seen that there are problems with getting metals from the environment. The second problem is that many metals are toxic to living things, including the metals that we need. So even essential metals are toxic. Even these metals that we think of as necessary, like copper and iron, for example, are toxic in large amounts. For example, the FDA says that iron in multivitamins is the leading cause of fatal poisoning in children under 6. Metal toxicity is a factor in a variety of diseases. Let's use copper, for example. Copper is an essential element, but it can also be toxic. There's a disease where copper is accumulated in the body called Wilson's disease. And you can see this copper accumulation in the form of these copper deposits in the eye. This is a picture of someone with blue eyes, and they have copper deposits around their iris. These copper deposits are not only seen in the eye, but they also cause liver damage and neurological damage. Symptoms of Wilson's disease are tremors and loss of memory. So we see here, copper is essential, but it can also be toxic. Another example is iron accumulation in the disease called hemochromatosis. You can see this person's skin is orange. The orange color is coming from iron deposits in their skin. Hemochromatosis causes heart and liver damage, pain, and arthritis-like symptoms. So once again, iron is essential, yet it can also be toxic at high levels. Getting it just right requires control. Even when we have the right amount of levels in our bodies, metal toxicity can arise from their ability to reduce oxygen. Redox active metals like copper and iron participate in dangerous free radical chemistry. This kind of chemistry can produce dangerous free radicals like the superoxide radical and the hydroxide radical. In this cycle, you can see that copper 1 can be reduced to copper 2. Redox active metals like copper can react with normal products of metabolism. In this scheme, copper 1 can react with the normal product of metabolism, hydrogen peroxide, to oxidize copper and produce a very dangerous free radical species, the hydroxide radical. The copper 2 can then be reduced by another normal product of metabolism, the superoxide radical. This is a free radical, but it's not very dangerous. And it gets oxidized to dioxygen, in the process producing copper 1. This copper 1 can react again with hydrogen peroxide, and this cycle can happen over and over again. Biological organisms need a way to regulate the amount of metal in the body, and they also need to be able to control this whatever metal is there. Biological systems need to keep metals in an inert or at least less reactive form so that they can be used advantageously and avoid harmful side reactions like this redox chemistry. Now let's look at an example of how biology has solved some of these problems. As mentioned on the last slide, metals can reduce oxygen and create free radicals that cause lots of damage. The take-home message is that you can't allow free metal ions to run around willy-nilly in cells. They must be carefully controlled or chaperoned so that they don't do any damage. Now, collecting iron from the environment is a big challenge due to its tendency to exist as rust or iron oxide and in very low concentrations in solution. Organisms like bacteria and fungi have developed beautiful ligand systems known as siderophores to scavenge for iron. These ligands have high affinity for iron 3. A prime example is the formation constant for enterobactin of 10 to the power of 49. That's a binding constant. So the K value for binding to iron is 10 to the 49. These organisms make these complex ligands and excrete them into the environment. Outside the cell, the siderophores complex iron and bind to the receptors on the cell surface to be transported into the cell. Once the metal-bound siderophore is transported back into the cell, the iron is released through chemistry and the organism can accumulate iron. Interestingly, these iron-binding ligands are also used to treat diseases like hemochromatosis or other iron overload diseases. In mammalian systems, transferrin is the iron that transports protein in the blood. There are 3 types of transferrins: lactoferritin, lactotransferrin, found in extracellular fluids like breast milk, and ovotransferrin, found in egg whites, are thought to provide an antimicrobial effect by competing with bacterial siderophores for iron. What I'm saying is that bacteria that are invading your body have to compete with these transferrin molecules in your blood. Your body produces these transferrin molecules to prevent the bacteria from getting the iron. So this is not only a way to transport iron in the blood, but also to keep it away from bacteria so that they cannot invade your system. If these proteins sequester the iron, the bacteria can't get enough and won't produce an infection. The third transferrin is serum transferrin. This is used to transfer iron in the blood to cells. It binds two iron 3 ions to prevent unwanted redox chemistry while the iron is in transport. The iron-loaded transferrin then binds to transferrin receptors on cell membranes where it is taken into the cell for iron release. Apotransferrin is transported back outside the cell to repeat the cycle. Once inside the cell, iron transport and storage proteins prevent unwanted redox chemistry from happening. The protein ferritin is used to store iron. This molecule has an outer coat of protein with an inner core of hydrous ferric oxide. Much like a Tootsie Roll Pop, where the candy coating outside is the protein and the ferric oxide is the Tootsie center, a single ferritin molecule can store up to 4,500 atoms of iron. Ferritin stores iron when it is in excess and then can release iron atoms as needed when there are shortages. Basically, this protein traps iron and stores it so that no unwanted redox chemistry can occur. The iron oxide crystals inside ferritin are very similar to that of ferrihydrite, which has a unit cell structure as shown in this slide. Now, I could teach an entire course just on the transport and storage of iron. In fact, each metal has its own unique set of molecules that are dedicated to metal acquisition, transport, storage, and incorporation of those metals into important protein binding sites. Think about it. This takes a lot of effort. Every single metal will have a different protein shuttling system, a different storage system, and a different chaperone system to incorporate these metals into the important sites. This is expensive. And you might ask, why bother? Why bother with these metal ions? The answer is that these metal ions are very valuable. They allow proteins to carry out reactions that would otherwise be impossible. Here's one example of how metal ions can help in biological systems to carry out chemistry. One major role that metal ions play is in the activation of water to make potent nucleophiles. This is the zinc center in carbonic anhydrase. Its job is to convert carbon dioxide into bicarbonate. And the role of zinc in this protein is to activate water to do some acid-base chemistry. You can see here the zinc is bound to a water molecule. It polarizes this water molecule to release hydrogen ion and produce a potent nucleophile. This hydroxide ion is now ready for attack. Carbon dioxide binds to the protein adjacent to the hydroxide ion, and then the zinc releases the hydroxide in nucleophilic attack of the carbon dioxide. At this point, bicarbonate is now produced, and simple ligand substitution will complete the catalytic cycle. Acid-base chemistry is one of the most important roles of metal ions in biological systems, but there are many, many other important roles. Another important role of metal ions in biological systems is in producing oxidation reduction chemistry by supplying a large variety of redox potentials. Transition metals adopt more than one stable oxidation state. For example, iron is stable in both a +2 and +3 oxidation state, and actually more recently it has been shown that the +4 state is biologically important in catalytic cycles. The iron 2-3 couple is at 0.77 volts versus a normal hydrogen electrode. If this potential was unchangeable, it would eliminate the usefulness of the two stable oxidation states. However, proteins, by changing the geometry of the iron, can actually modulate the potentials of the iron in the protein binding sites. To give you an idea of the range of redox potentials for metals in biology, here are some common iron centers, along with their range of redox potentials. The simplest tetrahedral iron-sulfur center, with all sulfurs coming from cysteine residues alone, can span a range of redox potentials of 100 millivolts. This potential is modulated by bond lengths of the iron-sulfur bonds and by the nature of the pocket in which the iron-sulfur center is bound. First, the bond length dependence. If the redoxin center was simply iron bound to 4 distinct sulfurs, the bond lengths would shorten as the iron center was oxidized from iron 2 to iron 3. Bound to a protein, these bonds require global changes in protein fold, resisting the oxidation and therefore modulating the potential. The potential is further affected by the nature of the solvent pocket. When charge is created or rearranged in a solvent, the solvent has to rearrange to accommodate the charge. Polar molecules shift to counter the charge. In a hydrophilic pocket, water molecules can take care of much of the charge, but in a hydrophobic pocket, the protein itself has to do the accommodating. Again, this modulates the redox potential. These two pieces are the inner sphere and outer sphere effects referred to in Marcus theory. By changing the geometry and size of the cluster, the same building blocks, iron, cysteine, inorganic sulfur, and other amino acids, iron-sulfur clusters span a range of redox potentials close to a volt compared to the iron 2+, 3+ couple with a potential of positive 0.77 volts. Iron also plays a role in centers other than iron-sulfur clusters. The most common iron site, other than iron-sulfur cluster, involves heme. Since the heme is a very rigid structure, most of the modulation and redox potential in these centers derives from the axial ligand, both the nature of the ligand and the bond length. So now we can see how metals take part in acid-base chemistry and also redox chemistry. Another really important use for metals are in structural roles. Now, as far as structure goes, you might ask, what can metals get you that carbon can't? And the answer is a variety of geometries. For carbon, you can only have 8 electrons, and so you can only have linear, trigonal planar, or tetrahedral geometries. With metal ions, you have much more variety of geometries that are possible, and much more coordination numbers from anywhere from 1 to 8. Here I've shown coordination numbers of 2 to 6 and what their geometries typically look like. The other thing that metals offer are flexibility and lability of structure. While certain metals do prefer particular geometries, coordinate covalent bonds are somewhat labile. This allows changes in bond length, substrate on-off binding, and even ligand exchange in response to or driving protein folding. And one of the best known examples of this, heme-bound iron in hemoglobin binds oxygen reversibly. And this is how oxygen is transported in your blood. Metals also offer scaffolding for protein folding. A zinc finger binding site is one very common example of this. In this type of protein, zinc acts as basically a structural glue for the protein. And without the zinc atom, the protein does not fold correctly. So in this way, zinc can act as a scaffolding for protein folding. Other metal ions do this as well. For example, calcium. This is calmodulin. Calmodulin is a calcium binding protein that only interacts with receptors after calcium is bound. Cell receptors will recognize the tertiary structure upon calcium binding. Notice that the tertiary structure is different after the calcium binds. In this way, calcium is acting as a structural scaffold for protein folding. Here's a closer look at a zinc finger binding site. Many transcription factors include these zinc finger binding sites in their DNA recognition domains. These are typically his2, cis2 sites, or basically they have 2 histidines and 2 cysteines that bind to zinc. Without the zinc, the protein does not fold correctly and can't bind to DNA. And once again, if zinc wasn't bound to this protein, it wouldn't fold correctly, and therefore it wouldn't be able to bind to DNA and make transcription happen, and therefore your cells wouldn't be able to express proteins correctly. So you can see here in this that there are amino acids binding to the zinc. So what are the biological ligands that bind to metal ions? And as you've seen, amino acids and proteins, side chains, are one of the most common biological ligands. Things like side chain groups, amides or oxygens that are part of the backbone, carboxylic acids, and amines that are part of the carboxy and amine terminal can all act as metal binding atoms. Small molecules like carbohydrates or porphyrins, like that we see in hemoglobin. DNA can also bind to metal ions. DNA bases can potentially bind, but one of the most common places for metals to bind is the phosphate backbone. In fact, magnesium binds very well to the phosphate backbone and actually is important for folding of DNA. Here's a table from your textbook that lists the 20 naturally occurring amino acids. The amino acids that are most typically found binding to and interacting with metal ions are highlighted in the pink boxes. These are the ones that you should know as metal binding amino acids. They're not the only metal binding amino acids, but they're the most common ones. The preference for any of these amino acids for a certain metal ion has nearly everything to do with hard-soft acid-base theory, with a small pinch of geometry thrown in. So when we talk about metal binding sites in proteins, you're going to be applying the things that we've learned before about coordination chemistry and preference based on hard-soft acid-base theory and geometry.